Namaste students, हमेशा खुश रहो बिटिया. I am JV'n's Dr. Neeraj Dholia, Faculty of Agriculture and Veterinary Science, Jayoti Vidhyapeeth Women's University, Jaipur. So today we are going to discuss the topic that is the recent development in product isolation. So this topic comes under the subject upstream and downstream process. So what are the recent developments in the product isolation process, that is the downstream process? So the development of techniques and methods for protein purification has been an essential prerequisite for many of the advancements made in the biotechnology. So the purification is not a new thing. The product purification is already a conventional thing, but the advancement in the biotechnology process make it easier and efficient than the previous ones. So the specifically the protein purification varies from simple one-step precipitation process to a large-scale validated product processes. So there are a variety of purification methods are there. Simple one-step precipitation method is also there, in which you add some reagent which creates the precipitation and you isolate the products by collecting the precipitate. So this is a simple one-step process in contrast to the large scale validated products processes. So that large scale validated production processes often have more than one purification steps and that are necessary to reach the desired purity. So if purity is not a concern, you can use the one-step precipitation processes. But if the purification method includes the purity, if the purity is required in the purification step, then you need more complex and the validated procedures. Okay, so for example, if I can say that the products of the molecular grades which are used in the research purposes have very high purity, and that cannot be achieved using the one-step process. So that may involve the multiple processes or multiple steps. So the key to successful and efficient protein purification is to select the most appropriate techniques, optimize their performance to suit the requirements, and combine them in a logical way to maximize yield and minimize the number of the steps. So the successful and efficient purification process have two things. One, the maximum yield, and second, the minimum steps. Maximum yield, why? Because in many special cases you have limited number of the host cells, like the bacteria, the animal cells. You have limited number and you want to get the maximum product out of them. Okay, so that is one thing. And second, the minimizing the steps, because as the steps are increasing, the cost of the product will increase, and simultaneously the chances of error and loss of the product is also there with the increasing number of these steps. By the way, the most purification schemes involve some form of the chromatographic techniques. Okay, in the earlier lectures, we have already studied about the various types of the chromatographic techniques. So, as a result of chromatography has become an essential tool in every laboratory where the protein purification is needed. So, various chromatographic techniques with different selectives can form powerful combinations for the purification of any biomolecule. So suppose if a biomolecule have a lesser purity in one chromatographic technique, then you can use the combination of chromatographic techniques, such as in the first step you can isolate the biomolecules using the ion exchange chromatography. Then you can use the elute further with the affinity chromatography. So more than one chromatographic techniques can also be used to increase the purity of the product. Simultaneously, the recombinant DNA technique is also helpful in the downstream processing or the advancements in the purification of the products, because what happened that in the natural cases, in the natural cells, the product which is you want to isolate are natural. They do not have any specific probe, any specific tags which help you to isolate them, to identify them. But with the help of the recombinant DNA techniques, you can modify your product in such a way that itself have the tags or the probes which help them to isolate or purify. Okay, so the recombinant DNA techniques revolutionize the production of proteins in large quantities. The recombinant proteins are often produced in forms which facilitate their subsequent chromatographic purification. Although the host contaminants are still present and the problems related to the solubility, structural integrity, and biological activity can still exist. Okay, after using the recombinant DNA techniques, there are some problems are still existing, like the host contaminants, means where the product is produced, the contaminants can be originated from there also. The host cell components is one of the most contaminant. So, although there may appear to be a great number of parameters to consider, with a few simple guidelines and application of the three-phase purification strategy, the process can be planned and performed simply and easily with only a basic knowledge of the detail of chromatography techniques. So, to achieve the good quality of the product, good purity of the product, and the efficient isolation of the product, you have to have the good knowledge of the various techniques such as the chromatographic techniques, so that you can use the perfect technique for the perfect product. So it is not a general concept that any product have specific techniques. Okay, it varies conditions to conditions, that what are the conditions that in which chromatographic technique give you the ultimate result or optimal result. So thank you. Hope you are satisfied with this detailed session, and if you have any query, please write in the comment box. I will resolve your query. This session is powered by digital version 2 Jyoti Vidhyapeeth Women's University Jaipur. Thank you.